Hello, I'm Caroline Leroy and I will be presenting to you microbiome maturation in early life and how we aim to define it and try to impact it to improve health of children. So as a disclaimer, I am an employee of the Société des Produits Nestlé in Switzerland and I worked at the Nestlé Institute of Health Sciences. I will start the presentation by giving you a quick overview of the gut microbiome in infancy. So the gut microbiome is defined as the ecosystem that resides of microbes that resides in the gut of human. And we have it that evolves throughout life with a specific period between birth and 4 years of age where we see a rapid evolution of its composition. So this is mostly characterized by an increase in alpha diversity that capture how diverse the ecosystem is. And you can see on the left-hand side of the slide a plot that shows the diversity of the microbiome between birth and 80 years of age. And you can see this sharp and rapid increase of this diversity earlier on in life that then stabilizes to remain at a certain level throughout life. On the second part of the slide, you can see that not only diversity changes rapidly during this early childhood, but also the composition of the microbiome, meaning the type of bacteria that are present in the gut, but also their relative abundance. And you can clearly see at 1, 2, 3, and 4 years old how this prevalence of bacteria evolves rapidly and then reach maturity, or something that we call being like adulthood microbiome and remain stable throughout life. So why are you interested in how this microbiome shape early on in life? Because we know that this is actually impacting host health and therefore the development of the children. And I will give you now a few examples of why we're interested in understanding how this happens and how this is linked to several health outcomes. So here, what we first start to describe is the environmental factors that can affect this microbiome maturation early on in life. And we know that things like the way babies are delivered and also antibiotic consumption are actually important environmental variables that will affect the development of the microbiome early in life. And again, in this left side of the plot, you can see a picture that shows the prevalence of certain bacteria in the gut of infant at 4, 7, and 21 days, and later on in infancy, so towards 1 year of age. On the left side, you have for infants that are vaginally born, and on the right side, you have an example for the infants that are born through C-section. And you can clearly see that the two pictures look very different. And this is mostly due to the fact that infants that are born through C-section actually have a lower level of bifidobacterium than the other group of infants. However, you can see that the picture stabilizes towards infancy and it's not possible to see any differences anymore. This is also clearly pictured on the right side of the slides, where you can see the evolution of bifidobacteria between 0 and 48 months of age, with in grey the normal infants, so the ones that are born vaginally and don't receive any medicine. And you can see this peak of bifidobacteria happening before 3 months of age. A similar picture is also observed for the C-section born infant, but you can see a delay in this peak of bifidobacteria. Finally, in blue, you can see the infants that receive antibiotic, and you clearly see that this impacts the prevalence of this bacteria in the gut of infants. Why do we think that this is important in terms of health? Because we know that C-section and antibiotic consumption are both linked to several risk factors in terms of health. First one being immune-related disease. As we can see on the first graph on the left, on the top on the left, that infants that are born through C-section are more likely to develop asthma later in life, and on the bottom graph we see exactly the same thing regarding antibiotic consumption. Second aspect is that also both C-section and antibiotic consumption put infants at risk of becoming overweight or obese later in life. Last example linking microbiome to health, you can see here that the scientists have looked at infants that develop bronchitis versus the ones that don't develop it. And what they observed is that the infants that develop bronchitis have lower level of bifidobacterium in their gut, and more specifically of Bilongum subspecies infantis. Besides the importance of the type of bacteria that are present and their relative abundance in the gut, what is also important to look at is what is their metabolic activity. So what are they doing within the gut of those infants? And something that we look particularly at are the short-chain fatty acids that are product of fermentation by the gut bacteria. And here what you can see is that when you compare short-chain fatty acids between infants that develop bronchitis versus the ones that don't in infancy, they have different levels of the short-chain fatty acid within their gut. So now the next question, knowing all those changes that are linked to health in infant, we want to know how best to capture those changes of the gut microbiome and its evolution in infancy. And the idea here, and what we're working on in the department, is to try to develop a measurement that would look like what is done for growth in childhood. So here you have an example of what happened for weight in a child between 0 and a few years of age, where you have a curve that the child is supposed to follow and to remain in an healthy bracket. And we want to see at different age where the child is on this weight range. And then, if you see that the child deviates, you can start to think on how can we intervene to try to help them to go back towards a normal weight range. We want to develop exactly the same idea, but this time for the microbiome, where you would have as the x-axis time, that would be the age of the children, and as the y-axis, something that we will call this early life microbiome maturation, that will capture how the microbiome evolves throughout time. And the idea is really to develop this curve as it was done for weight, to see how we can support the maturation of the microbiome. Also help to identify how risk factors impact this microbiome maturation and make infants deviate from it, and how we can help them to normalize back towards a normal trajectory through things like nutritional interventions. However, this is relatively challenging because, as we saw before, the microbiome evolved in a very complex way. We did see that indeed there is an increase in the diversity of the microbiome between birth and 3 years of age, but that there is also this sequential succession of microbes that will come and colonize the gut, with some of the bacteria reducing their prevalence with time and some others becoming more and more prevalent. Finally, we also saw that not only the bacteria that are present in the gut are important, but also what they are doing, their metabolic activity and their functions. So to create one trajectory is quite tricky because we need to capture all those factors that are important for the development of the child together. So far what researchers have done is they've used information at the bacteria level to try to build this type of trajectory. And here what you see is a successful example that was published in 2015, where they've used machine learning approach to generate a trajectory that captures the microbiome age of the child, so how mature the microbiome should be at a certain age. What is interesting using this type of approach is that once you've built your trajectory to define what should be the range where your microbiome should be at a certain age, you can then look at which bacteria are actually important in building the trajectory. And here you can see the ranking of the most important bacteria that were used to build this example of trajectory, with, for instance, F. prosnitzi being the top bacteria coming in the model. However, all this is quite challenging. The first thing we need to define if we want to develop this healthy microbiome trajectory is what is actually healthy. So which kits do we include when we build the trajectory? We saw before that there are indeed several factors that we know can impact the microbiome and its development in early life. So there are things that need to be considered, such as breastfeeding, mode of delivery, adverse event, or medication use. Second aspect is how can we standardize this procedure? How can we make a trajectory that can enable us to compare different studies to one another on different clinical trials, and also be relevant to different geography, because we know that the microbiome, depending on where you're born, will be different. And actually this task is not easy because there are a limited number of studies that have looked at microbiome between 0 and 3 years of age, and you can actually see that on this pie chart that is depicted in yellow. And it's less than quarter of the studies so far on the microbiome that are conducted in this age range, meaning that we need more data to be able to construct those standardized approach for microbiome maturation trajectory. Finally, the last thing that is important is to know how relevant this trajectory can be in understanding how risk factors affect the microbiome maturation and how this is linked to health, and also how diet can impact the trajectory. For the last part of the talk, I will be talking about diet and how we can use it as a means to actually influence the gut microbiome maturation. So I will start with showing you some evidence that diet is indeed a good way to impact the gut microbiome development. And I will start with these first studies that we already discussed, looking at vaginally versus C-section born infant and how they differ in terms of microbiome composition. What the researcher also did here is to collect a certain number of metadata and information on those child and try to see how those information could be relevant in explaining the composition of their gut microbiome at 4 days, 7 days, 21 days, and then after 1 year old. And what they identified is that breastfeeding, that is in the square in red, was actually an important variable in explaining how the composition of the gut microbiome of those infants was. Here is a second graph that shows a very similar picture, where this time they looked at 800 infants in the US and EU at different age, so you can see all the age being pictured from left to right, and they also observed that breastfeeding was an important variable and even the most important variable between 3 and 6 years old in influencing the composition of the gut microbiome of this infant. The second aspect beside breastfeeding was that solid food also appeared as an important element in determining how the microbiome was shaped in those infants. There are also some studies that have looked deeper in solid food in trying to understand which components are actually important in shaping the gut microbiome composition, and you have an example here of a study that was conducted in 48 infants. So this is a PICO-A plot where all the dots will be one sample, one microbiome sample, and the more the dots will be close to one another, the more similar the microbiome composition is, and the more distance, the more dissimilar. And the gradient here shows the age of the kid, with the white dot being younger kids and the red ones being older kids. And then what they did was to see if they could use solid food intake as a way to explain these changes in terms of microbiome composition. And they did show that, for instance, the kids that are, or the samples that are at the bottom part of the plot, our microbiomes are more linked to fruits and vegetable consumption, while the ones that are on the top of the plot will be more linked to other roots and vegetable consumption. So really showing the importance of solid food in shaping the composition of the gut microbiome. But how all that is relevant to our trajectory, and is the trajectory actually a good way to observe the changing of diet on the microbiome? So if we go back to the studies that we showed earlier with this initial trajectory using bacteria to develop it, what they also did was an intervention study, a dietary intervention study. And this was done in infants that suffered from severe malnutrition. And they randomized them into 2 different arms. And what is interesting here is that they then put those, the samples that they collected in those infants before and after the intervention on these trajectories that they defined earlier in the study. And what you can see here on this first graph is the fact that infants that suffer from severe acute malnutrition, for their microbiome, it's actually placed below this trajectory, showing that clearly the trajectory is capable of discriminating the fact that they have a condition and therefore a different microbiome maturation. They then looked at their microbiome sample following the intervention, and what they showed is that this was able to redirect the kids towards the initial trajectory, meaning that the trajectory is actually a good way to see how risk factor can influence microbiome maturation, and it can also help us to capture how a dietary intervention can help the kids going back towards a more normal microbiome trajectory. Okay, so we saw that diet can influence the maturation of the microbiome. However, there are several challenges that need to be considered before we move forward. And here what we wanted to represent is actually the fact that we know that diet influences microbiome, but we don't know to which extent. And what you have pictured is in a study in adults, they looked at the different environmental variables and their effect on microbiome composition, and in orange you have the average weight of diet impact on the composition of the gut microbiome. And you can see that actually this is not that much. It's less than 10% of the variance of the microbiome in the population that can be explained by diet. And this is mostly due to the fact that we don't capture diet maybe in a way that is optimal to be able to characterize its effect on the microbiome. And this is better pictured in the study here that was also conducted in adults, but it's a nice example to show how we need to change the way we may look at diet to understand its impact on the microbiome. Because diet traditionally is mostly looked at in terms of nutrient intake. And this is actually what is shown on the figure F of this slide, where what they did was to try to see how dietary intake could mirror the composition of the microbiome in adults. And what you have pictured is that in purple you have the microbiome sample and its composition, and in blue it's linked to the nutrient intake. And what they show is that by trying to do that, they were not able to explain microbiome composition with what people were consuming in terms of nutrients. However, what they did is to repeat exactly the same type of analysis, but this time not looking at nutrient intake, but food in general, so whole food intake. And you can see that in the E figure of the graph, and they were able to show that it was a much better correspondence between microbiome composition and dietary intake. They also repeated that, but this time focusing on grains and fruit intake, and the same, they were much better able to explain microbiome composition with dietary intake, looking only this time at grains and fruit. So really that shows us that when we look at diet, we need to reconsider the way we do it from a microbiome perspective, because they may not be interested in the same food that we eat than us. For instance, what is important for the microbiome will be the fiber content, because that goes up undigested towards the end of the digestive tract and will therefore feed our microbes. There are other elements such as polyphenols that also appear to be important and that are not traditionally used when we look at diet. And this is the type of things that we are working on to try to see how we can influence microbiome and its development in early age. So now a few take-home messages. What we saw is that there are several windows at which the microbiome develops differently and can be actually impacted by risk factors such as C-section, antibiotic, or adverse events. And that those are occasions to actually help infants developing their microbiome, and we can do that through diet. So we've seen that breastfeeding is actually an important helper in terms of microbiome development, and then after the introduction of solid food and diet in general can also influence the development of this microbiome, and this gives us several windows of opportunity to do that. And with that, thank you for your attention, and thanks to the rest of the team that worked here.